Hey guys, so today I'm going to be talking to you about the citric acid cycle. I am actually in love with the citric acid cycle because before this project I did not get it whatsoever. It was the hardest thing in the world, but now it's so simple and I hope you find this video helpful. Basically, all the citric acid cycle does is takes the energy from food and makes it into a usable form that our cells can actually use. Now, before the citric acid cycle takes place, there's a thing called glycolysis that happens beforehand. Don't let that scare you. All that glycolysis is the breaking down of glucose by enzymes. This releases energy and also gives us pyruvic acid. The pyruvic acid is nothing scary either. It basically is used in many metabolic processes. Alright guys, so this is going to be basically the entryway into the citric acid cycle. What's going to happen is enzymes are going to break a CO2 off of this pyruvic here. So a carbon dioxide is going to be broken off of this 3-carbon chain. This is going to yield an acetyl-CoA right here, and it's also going to yield an NADH that's going to be right over here. Acetyl-CoA carries 2 carbons and fuels the Krebs cycle. With its energized electrons. It is the chemical energy from food, and it transforms it into other forms your cells can use. Alright guys, so at the start of the citric acid cycle, we're going to have our acetyl-CoA right here. So basically acetyl-CoA has 2 carbons that it carries. They actually get ripped away by some enzymes. These enzymes give the 2 carbon atoms here to oxaloacetate right here, combining, making a 6-carbon chain that is then going to be our citric acid. In this isocitrate step, what's going to be happening is the NAD plus right here gains an electron carrying function, which becomes an NADH. NADH goes to the electron transport carrier chain, which in the respiration process powers ATP creation. Now remember, the electron transport chain does not happen in the cycle, it's actually going to happen after the cycle. After the isocitrate that yielded the NADH comes a 5-carbon chain known as alpha ketoglutarate, which is going to be right down here. Now here we're actually going to be getting another NADH. From the NAD plus oxidation, we now are reduced to a 4-carbon molecule called succinyl-CoA. Succinyl-CoA has such a high amount of energy still that enzymes can still use this. Here we get GTP from GDP, and we get ATP from ADP. GTP right here is an energy-containing molecule and is an additional product of this cycle. We also get 1 ATP, which is really exciting because remember, ATP is what powers the cells. So this is basically a monumental process in this whole cycle. Now there's enough energy for enzymes to reduce, so we're actually going to be getting the succinate. From succinate, we're actually going to be getting an FADH2 from the FAD. So as the succinate gets oxidized, FAD gets reduced and gives us FADH2 like I mentioned previously. 1 more NAD plus will get reduced right up here. 1 more NAD plus is going to get reduced to give us an NADH. Remember the oxaloacetate that came in at the start of the citric acid cycle? Well, we have oxaloacetate at the final stage as well. That's going to be right up here. And it is so eager and ready to meet acetyl-CoA again. For every glucose cells absorb, the cycle runs. It's going to be running 2 times. The Krebs cycle is continuously running, but it can only run as long as the cells get fuel. So in other words, we have to eat. And as a result, we exhale all the carbon dioxide that got released from this cycle.